what attracted me to CERo is the platform, and I'll describe what we have and what we're hoping to accomplish. So CERo is based off of the acronym CER, which stands for a chimeric engulfment receptor. So of course it's a play on a chimeric antigen receptor terminology. What chimeric engulfment receptors are, chimeric engulfment receptor T-cells, are autologous T-cells that have been re-engineered with the CER, which essentially comprises a binding domain that is derived from myeloid lineage cells. And so these are natural receptors that are involved in identifying and removing through normal homeostatic mechanisms diseased or dying normal cells, right? So every day in the body, particularly with your immune system, you have a big turnover of dying immune cells that need to be identified and cleared from the body. And so there are natural class of receptors that macrophages and mature dendritic cells express that perform this function. So it turns out you can actually take these receptors and then fuse them onto intracellular signaling domains and do this in a variety of different ways to impart a new biology into T cells. And so what we're doing at CERo is taking these phagocytic receptors, we'll call them, putting them onto different intracellular signaling domain backbones that contain both T cell and myeloid related cell intracellular domains that lead to a couple of different things. One is CAR-like activity. So you can reprogram a T cell to go after a common target. In our case, these are drug-inducible stress proteins that can be brought up through targeted small molecules that are being currently used in a variety of different oncology or across different oncology indications. And get these cells to then behave like CARs. So we see cytolytic activity, cytokine production, proliferation. But more importantly, with the engulfment receptor and using myeloid-derived intracellular signaling domains, we're actually showing that we can enhance the antigen presentation-like capability of a T cell to try to drive up both class 1 and class 2 tumor antigen presentation. So you can think of this along the lines of a macrophage, but in a T cell. So we have cytolytic function going after a specific target that leads to some percentage of those tumor cells that are being engaged, being engulfed by the CER T-cells, and then antigens being presented back to the immune system. So the hope is through this process, we can not only get to CER-mediated tumor clearance, but hopefully stimulate what we refer to internally as a secondary immune response. So potentially bringing in other endogenous machinery that the patient has, NK cells, myeloid-derived cells that are favorable to help with tumor clearance. And of course, if we can get some epitope spreading from the CER T-cell product, we may be able to boost further the T-cell-based immunity towards a patient's tumor. So this conceptually then allows us to go into a variety of indications, leukemia, lymphoma, of course, though it's a very crowded space. But we think more importantly, we can use this technology in the setting of solid tumors. So we can take CAR-like function with this chimeric engulfment receptor-like function or enhanced APC capability to allow us to specifically target tumors in combinations with small molecule type drugs, a standard of care that the patients are already on, and hopefully tackle metastatic disease through this epitope spreading secondary immune response mechanism. So it's quite complicated. We've got a long ways to go, both preclinically and then, of course, in the clinic to prove all of this. But I believe the technology is there, and we have a great team at CERo that will try to advance to an IND, hopefully end of next year, if not early the following. But we're moving along pretty quickly in that regard.